Hello and good evening, everyone. Thank you so much for joining us for our very first Alumni University. We are so excited to have Britt Burton-Freeman joining us this evening. For those of you who don't know me, I'm Kristen Kipke. I'm the Director of Constituent Engagement here at Illinois Tech. And my colleague Meg Munch is also joining us behind the scenes. She will be running the webinar platform. So just before we get started, a few housekeeping items. The Q&A section will be disabled during this webinar. So if you have any questions, either during Britt's presentation or at the end, please feel free to type them into the chat box at the bottom of your screen. And Meg and I will go back and forth and make sure that all of your questions are answered. So to introduce our presenter, Britt Burton-Freeman, Ph.D., is the director of the Center for Nutrition Research. She's also chair of the Department of Food Science and Nutrition and a professor of food science and nutrition here at Illinois Tech. She received her Ph.D. from the University of California, Davis, her Master's of Science at University of California, Davis, and her undergraduate degree from the University of California at Chico. Her research here at Illinois Tech is multidisciplinary, demonstrating the strong links between the sciences of food, nutrition, food chemistry, biochemistry, physiology, microbiology, pharmacology, and behavior to understand how diet and dietary components impact human health during normal, stressed, and pathophysiological states. And with that, I will turn it over to Britt to begin her presentation.
All right, well, thank you so much, Kristen, for the introduction. I'll try to share my presentation here and we'll get started. Can everybody see this okay? I hope. I actually can't see you to give me the thumbs up, but I will trust Kristen or Megan to let me know if there's any technical difficulties. If you can't hear me or any other challenges that you may encounter along the way.
Things look great, Britt.
Hello?
Things look great, Britt.
Oh, good. Okay, good. All right, so I'm going to talk about fermenting for life. And this is a fun topic on a cold, dreary night. But I think that we'll get a lot of life and discussion, hopefully at the end. It sounds like that's going to be the format as I'll present. However, if you have a question along the way, I believe we discussed that Kristen or Megan would field them at a point that it made sense in the presentation. So, really, I want this to be casual and interactive if possible. In fact, I'm realizing now that I do have some interactive slides. So we'll see how this works out in a webinar Zoom series. So I thought it might be helpful to start out with just a little bit about me, although I'm not getting this to move to advance. Oh, let's see, I have to do this. Here we go. So Kristen gave an introduction. I am from California. I was born and raised in California. I did all my schooling in California. But I'm in food science and nutrition, I think, in part because I grew up with food. I was born to a walnut farmer, and that kind of serves as the food and agricultural side of me. And then my mom was in medicine, and so there's the health aspect. So I'm a nice mix between my parents. When I give presentations to students that are going to be joining our program, or even students that are in the alumni status and are maybe rethinking, what are all the different opportunities. My degree in nutritional biology took me to my first job, not in a traditional nutrition position, but rather at a biotech R&D center. I worked for Amgen, and I went there because of my physiological background in obesity and metabolic disorders. They were looking to develop a drug as an anti-obesity agent, and they told me, we can teach you drug development, but we can't teach you what you already know in metabolic disorders. And so anyway, it was an amazing experience, and you never really realize where, what you know or where it will be applied. So We're definitely cross-cutting. So as Kristen was reading off some of the different areas that we touch, indeed, food science and nutrition is a multidisciplinary science in and of itself. It really integrates a lot of the other sciences. When I'm not working, I have a beautiful family, husband, 3 kids. We do a lot of outside activities together, turkey trot and more. But I also spend a lot of time with students. I love to do student outings and work in professional societies. So I'm very blessed to do a lot of what I just absolutely love. Here at Illinois Tech, we have a research center, the Center for Nutrition Research, and it's really focused on preventing and managing disease through diet. Here again, I would say I'm blessed to have a research team that is, excellent in every bit of the science that they do, results oriented, but really quality results is what they're focused on. So I do lead the Nutrition Research Center, but I do it with very strong collaborations. In fact, I feel like I co-direct with Dr. Edisering and Sandu. I thought it might be, important to share some of our capability. A lot of people don't know what we have here at Illinois Tech, but we run clinical trials. So we have a human research center. We have sophisticated biochemistry and analytical chemistry labs. Some of the microbe work that we do, not only in food safety, but in the microbiome, understanding the role of the microbiome in health and disease, which is pertinent in fermentation and the health benefits of fermented products can be done right here on our campus, one of our campuses, the Moffitt campus. We do imaging studies, and we have a brain lab that we do cognitive and behavioral functions in the tower. And then we do a number of mechanism of action studies. So in all, really, if you kind of look at our research, it's, kind of looking at the intersections of these various diseases, but it's the diet, gut, brain axis that we study. And so anyway, it's a great thing to be working on any day and age, but particularly this day and age. And our impact, our research impact hits a number of different areas: nutrition policy, medical science and discovery, the food and ag sectors. But proudly, we can give a distinctive education and research opportunity to Illinois Tech students to set them up to be leaders, future leaders in a number of different areas of their interest. So with that, just brief introduction, we'll move into fermentation. Fermentation is an exciting area. It has just absolutely been booming over the last 5-10 years. There's more and more that we're going to learn as we go. Much of it in the past has been focused on preservation, but nowadays it's got a whole new slant, and I'm going to try to capture some of that while also laying the foundation of, what is fermentation. And so some questions on your mind, and maybe why you joined today or this evening is, what constitutes fermentation? How is it defined? Do fermented foods necessarily contain live microorganisms? And if they don't, are they still considered a fermented food? Are fermented foods the same as a probiotic food? Is there a difference relative to pickling and fermenting? Do microorganisms in fermented foods become established in the gut or influence the gut microbiota? And do fermented foods provide health benefits? And if so, how do they do it? So hopefully what we'll do through the next half hour is we will cover some of these or all of these topics. And I apologize, but my Siri phone that's sitting here seems to think I'm talking to her. So I thought it was important for me to maybe keep time, because I can really get going and lose track of time. All right, so fermentation. Fermentation stems from the Latin word fermentare, defined as to leaven. The practice of fermentation is historically rooted in cultural norms, mainly for preservation purposes, but also for flavor. I did a little other, there's other bits of reading that says that, maybe even really early times, it was before the persistence of the lactase enzyme. And so it was to be able to consume dairy products. And fermentation certainly takes care of the lactose component that can give people trouble if they don't have the lactase enzyme. So there's been a number of reasons to ferment foods in the past, but more recently, we've become interested in fermenting because of the culinary distinctions. And so, this is really starting to differentiate products, foods as well as beverages. And then the health benefits. I believe that probably one of the biggest, I'll call it discoveries, although we did know that the microbiome of our lower bowel existed, we just really didn't think it did much. But upon the discovery of these microbes in our lower bowel and how they interact with the host genome has really just, created so much research in the last, 5-10 years, that it has also given life to areas in food product development around fermentation, and fermentation as a process that, again, with historical, reasons and why it was used, but now has, some additional reasons. And culinary distinction and health benefits is one of them. So just from a broad speaking definition, we think of, or we define it as the transformation of food by various bacteria, fungi, and the enzymes they produce, but it also includes yeast. So there's a number of microbes that you might think of. And that was, Katz put that out as a definition, but just this month, Maria Marco was a lead author, and she was basically, her and a number of others were leading a panel around fermented products and really, what would be a better definition. And so this just came out: foods made through desired microbial growth and enzymatic conversions of food components. And the idea here is that it's more inclusive and more broad. But I think the distinction here is that the enzymes that are going to be doing the alterations are from the microorganisms themselves. And they're not enzymes from, say, the plant or from the animal product, but it's actually coming from those microorganisms. And fermentation, of course, if we're going to eat it, humans want things that taste and smell good. And so there's a bit of an art in there for sure. So factors influencing what foods and ingredients are fermented. There's a whole history with this as well. Cultural and social influences, traditions have a lot a large role to play here. Availability of food sources, super important. We just think about, regions of the world where dairy is predominant. And so we're going to have much more of these fermented products from dairy food sources, or dairy as a food source than maybe some other areas. Or in Korea, for example, kimchi and the use of radishes. Sauerkraut, cabbage. One of the findings is that we think of sauerkraut being, product from Germany, but some of the readings tell us that no, in fact, it really came from China. So anyway, lots of fun history. Taste preferences, raw materials, what bacterial strains are available. When we think about sourdough bread, that has some fun stories related to it, why sourdough breads might taste different. But environmental conditions, whether it's a dry climate or a humid climate, can also make a difference in what sorts of foods would be fermented. There's been technological development, as one might expect. One just might be in plant efficiency. So, what can we do to reduce fermentation time, get more products out there? But there's also been a number of innovations, and then we can just think about corn and biofuel. For those of you interested in the market, this is just, a highlight of that. You can see that global fermentation ingredients market accounted for about 29 billion in 2017, and it's projected to grow to almost 70 billion by 2026. So that's in 5 years. And so if you look at the pie chart there on the left or on the right, you'll see the The purple kind of violet color, and that represents just the food and beverage applications. And it's estimated that there's about 5,000 fermented foods and beverages produced and consumed globally. Much of the demand that is projected is estimated to be due to this segment. And it's, you know, the growing interest in having foods that provide a health benefit is what's, you know, really, I guess, underscoring this movement. But there's a lot of other things to be thinking about. There's huge expenses of ingredients and there's potential health risks. So it's not one of those things that there's no risk. In fact, there's also a whole regulatory component that we need to be thinking about, because that's not well established yet either. And much of that has to do with the health risk benefit. So types and categories of fermented foods. So, you know, there's different types of fermented products. Some are very dialed in. There's an intentional application of microbes, and there's even sequencing of microbes that are now patented for their specific flavor that they impart, say, for a beer. You know, so some of the artisan beers might have very specific yeasts that they utilize to capture that flavor. Same with wine. So this is an intentional application of microbes, very dialed in. A lot of the experimentation happens as maybe there's a new line that there's interest in developing. There's controlled, but a little bit wild fermentation. So kimchi and sauerkraut are good examples of this. Fermentation is allowed to happen, but it's controlled. So it's under certain conditions and it's definitely controlled. But the microbes are not intentionally added. So this is an example where the microbes from the plant itself are utilized as the starter, if you will. So it's kind of considered wild, but there is some control over the conditions. And if we think about sauerkraut or kimchi in a jar, one of the things that the developer will do is, add, some water, for example, but also add salt so it can control what bacteria will grow under a salt condition. And then certainly one of the things that we know about fermentation microbials is that as they grow, they crowd out the pathogenic ones. And then as the acid or the pH drops in the sauerkraut, for example, that will also keep some of those harmful bacteria from growing. Wild fermentation, it's kind of just that, wild, like I described previously. But in addition to microbes that might come off the plant, we might use yeast, for example, from the air to initiate the fermentation process. And so sourdough bread might be an example of that. So yeast from the air to get a starter culture going really might be the flavor differential between sourdough bread, for example, in New York versus sourdough bread made in San Francisco. So that just gives you a little sense of the different types or categories of fermented foods. There's also this other differential that I think is a bit fun to think about, and that is, when does fermentation take place? There's a fermentation process that can happen in the preparation phase of a product, or it can happen in the end phase. So the preparation phase is usually to accentuate a flavor profile or to achieve it, and then to achieve this aroma quality that you're looking for. Other steps after fermentation would be completed before finalization of the product. Whereas an end phase fermentation would be the fermentation that really is the end step. The end step may be that there's a pasteurization after, so it clearly stops it, or there's a control of the microbes that are doing the fermentation, and that can be modified by the feed, if you will. So I was kind of hoping to play a little game here, but since I don't think I'm going to be able to hear you, but Kristen, let me know if I can hear people. But I thought what we would do is we would look at this list, and if you look at these very common fermented foods, do they go on the left side, the fermentation in the preparation phase, or are they fermentation in the end phase? So if you were going to take these foods, so maybe if you just kind of want to think about it, and then I'll move to the next slide and give you the results, but which ones would go on the left side and which foods would go on the right side? So would yogurt be an example of a food where fermentation happens in the preparation phase, or is yogurt an example of a food that's fermented in the end phase? So bringing it to the final product. Likewise, coffee, kimchi, vinegar, soybeans or soybean products, chocolate, kombucha, beer, sourdough bread, wine, fermented tea leaves. So which direction do we go? Well, if you're ready for the answer, maybe put up your hand. Here we go. So this is how they fall out. Coffee, chocolate, fermented tea leaves, and sourdough bread, part of the prep phase, where all these other, I think I have 7 examples here. Fermentation is part of the end phase and getting to the final product. So then this brings me to fermented foods. Which ones have live microorganisms present and which ones have live microorganisms absent? And what makes the difference there? And which foods that we might think are fermented, but they're not fermented? And so on the left-hand side, you can see here that yogurt, sour cream, kefir, most cheeses, and many of these products, you'll see it will say live and active cultures. Okay, so it's still growing. Okay, but it's being controlled because it's usually these things are found in a refrigerated section. Okay, the coolness keeps everything controlled. If a product has been heat treated, so pasteurized, for example, that will essentially kill the live microbes. And in fact, one of the things that's going on in the kombucha world is that there's kind of some very interesting stories, maybe not some great ones, but they were still live and active on the shelf, and some were exploding. And so one of the things that they've had to do is kind of rethink how kombuchas are made and how they're stored. And pasteurization became a means to be able to control that. However, one of the beauties of kombucha is to have not only some of the microbials present, or not, but some of the bioactive components. So anyway, that's an area where there's a lot of development going on. The not fermented, like chemically leavened bread, here's where baking powder, that's a chemically leavened bread. So when we're making various breads using that. Pickling, if we're pickling, we're, for example, cucumbers and vinegar and salt and maybe some onions and some other spices, that is not fermentation, that's pickling. But there are ways in which you could actually achieve a low acid vinegar-like liquid in which you could have fermented pickles. So you would find those in your refrigerator section in your grocery market. Okay, so moving on to health benefits of fermented foods and beverages. So I think I mentioned at the beginning that as our knowledge has grown about the microbes of our large intestine, so has our interest in understanding how we can modify it. And so for a long time, we've known about probiotics and maybe some of the health benefits of consuming a particular microbial or consuming yogurt, but it really has been only recently that we've been able to start annotating this and really understanding what microbes are present before and after a live microbial beverage or food is consumed. Do they act as probiotics or vehicles of a probiotic solution, or are there other things that are going on? So this next section, I just thought we'll touch as much as we can about the health benefits of fermented foods and beverages and how they might do what they do. So one of the things that fermentation does is that it creates a chemical change of the food. So we talked about that. And when it does this or creates this chemical change through the enzymatic activity of the microbes, it can liberate or make more bioavailable compounds that have biological activity when they're consumed. And one of those types of compounds are phenolic compounds. There's been a lot of study about phenolic compounds in tea, for example, or phenolic compounds in berries. In fact, that's one of the areas that I do a lot of research is in the polyphenolic compounds of berries and berry products. And when we think about making these bioactive components more bioavailable, then we can start understanding how they might be interacting with different cells around the body and maybe even having a health benefit. But there's other bioactive compounds that might become more bioavailable, including essential vitamins and minerals, peptides that are formed. This is actually really important as it relates to some of the health benefits of some dairy products, the peptides that are made and its relationship to blood pressure regulation. Different sugars and fatty acids can also be modulators in our physiology. And so it's how these compounds are then absorbed and can have biological activity impacting physiological processes that are important as it relates to obesity management, which might include appetite regulation, reducing the risk of type 2 diabetes, reducing the risk for cardiovascular disease. So perhaps having an impact on the risk factors such as cholesterol. So if one has high cholesterol, how it might impact people that have high blood pressure or a propensity to have high blood pressure. And then there's also some work that's being done relative to cancer and the types of mutations and inhibiting those mutations. So the benefits could be direct or indirect. So one thing that we think about relative to a direct interaction is the ingestion of the live microbes, okay? So we're thinking about those products that are delivering live microbials that then may make it to our large bowel that will then help define and maybe modify the microbial population of our large bowel. And why would that be important? Well, one of the things that we're learning, and we're learning more and more every day, is that that composition derives an incredible function of the gut. And that gut is highly innervated neuronally, but importantly, our immune system. We have more immune cells or immune fighting activity in our gut than we do anyplace else, because it makes sense that that's the barrier, right? Before things get inside. So whatever we can take care of before it ever penetrates our intestinal tract and we can excrete, the better off we are. So a lot of immune function is driven at the level of the gut. And our microbes there have a huge role to play in that. And so modifying that composition and modifying their function is an important part of systemic health. So how that impacts the rest of our body. The indirect benefit is as a result of ingestion of microbial metabolites that are synthesized during fermentation. So this gets back to the bioactive compounds that are made. So it may be that they're bioavailable, but there also may be some metabolite changes. So the big compounds becoming smaller compounds so that they can be absorbed. All right, so what sort of health benefits? Gut health, I already described a little bit about gut health. A lot of it has to do with the immune system being anti-inflammatory, also making sure that those junctions between cells of the intestinal tract are nice and tight so that there's not a lot of permeability in your GI tract. There's suggested evidence for antihypertensive, so blood pressure lowering effects, antimicrobial, so helping to suppress pathogenic microbes and increase the healthy microbes that are in your gut. Immunomodulatory, so that's the immune system, anti-cancer, anti-thrombotic, so having activities relative to how sticky our blood is. Opioid activity, antioxidant, that's a very popular and well-known word. So helping to maintain a low oxidative stress, because oxidative stress that builds up can create damage, and that's where some of the cancer piece comes into play. But one of the biggest issues we have right now is that it's a relatively new field. So while fermentation itself is, thousands of years behind it, its demonstration of health benefits is extremely limited. And we do this by the clinical trials that we conduct. And so we know how that works. We know how it works in the drug industry. We're quite aware of how it works as it relates to vaccines these days. So the next piece here is that, well, if fermented food products or fermented beverage products have these health benefits, the ones that are claimed here, we need to have those demonstration studies. And so that's one of the things that's very exciting to me and graduate students in our lab, because they want to study the health benefits and put the science behind the health benefits that they deliver. And we can do that because we have a clinical nutrition research center. We can actually do this sort of research. Some areas of research or some areas in fermented products have gotten a lot of attention. So some of the dairy products, there's a lot more data out there, not only from a preclinical animal-based study, but there's more humans. But some of the newer products like kombucha, there's very, very little. Kefir even doesn't really have too much, but there's a whole area in which we can explore. And why does it seem like the science is behind? Well, it's only been recently that the technology to identify compounds pre and post fermentation has been available. So all the omics technologies, I mean, there's been a boom, a technological boom that has allowed us to do the sorts of research that we can do today. And that's one of the things that we're quite excited about, is because we have access to that in our labs, both in the tower on the Meese campus, as well as at the Moffitt campus. And we can study those direct and indirect effects of fermented products. So let's just take a few examples. I want to check my time here. I think we're doing pretty good. Going back to our list of those products that are fermented in the prep phase versus those in the end phase, if we think about, fermenting coffee, one of the main reasons that we do that is to get ready for the processing of coffee, really. But the fermentation is part of the process. It starts it. But what's really getting done here is we're ridding of the mucilages and letting the beans be ready for roasting. But the fermentation process also has a role in adding flavor. So does it have a role in the phenolic components of coffee? We know that coffee is loaded in phenolic components that have biological activity. That's an area that's not completely clear. What about other beverages like tea? Tea's been around forever. Tea leaves, we know that there's a number of phenolic compounds, but what about kombucha? What do these two things have in common? Well, tea, right? So on one hand, we have tea, we have black and green tea. And in fact, black tea is a fermented tea. So it's the black fermented tea leaf. And we know that there's these flavanol compounds and catechins. They have some very obvious structures that those in the field are quite familiar with. And many of these structures, because of the hydroxyl groups, have antioxidant properties. And much of that comes from these catechin structures. Kombucha, on the other hand, is tea. Okay, so we take black or green tea, but we add this symbiotic culture of bacteria and yeast. So they say adding a SCOBY. And it actually looks like, if you can see in this little jar, it looks like a little mushroom. It's kind of a fun thing. You might Google it, look at a YouTube or something, but it's really fun to see how you can have a mature SCOBY and how you can develop a baby SCOBY, and then how you keep them alive so that you can continue to make kombucha at home. course, there is, hazard, as there's hazard in anything. And so, really following the steps is important, because contamination can certainly wreak havoc. But the benefits here is that these microbes, the bacteria and the yeast, can convert some of those more complex polyphenols, so poly meaning multiple phenols that are connected together, into smaller phenolic compounds that are more bioavailable. And these can have the antioxidant effects that we talked about. They can have anti-inflammatory effects. But then the extra benefit that maybe kombucha can offer is these live microbials. So the microbial count increases. So you kind of get the double whammy, if you will. How about soy products versus dairy products? Dairy, forefront of probiotic delivery. I mean, we think about yogurt and kefir, abundant probiotics. So usually lots of different live and active cultures in a dairy product. Some of the peptides that are generated, because they'll utilize those, we kind of think about probiotics or these live microbials just working or utilizing sugar, but they also have their own nutritional needs, and some will need to utilize various amino acids. And as they take apart certain proteins, they will leave peptides behind that can be absorbed and have biological activity. Some of that activity is what's responsible for the antihypertensive effects that have been documented. Gut health, certainly the live microbials, and also some of the anti-inflammatory peptides that are generated. Soy is known for a number of flavanol type or flavonoid type compounds. These are isoflavones, and the proteins. Soy milks, some people say that the flavor is a bit, undesirable. And so what role can fermentation play in this? To improve flavor, for sure, right? Usually fewer probiotics, live microbials. So another word for probiotics is live microbials. In fact, that's the regulatory lingo, is to call them live microbial or live microbes. So there's fewer of those in dairy, in soy compared to dairy. But soy is an alternative delivery mechanism for people that can't consume dairy products. And again, some of the effects that have been shown: antioxidant type effects, hypolipidemic effects, and specifically lowering people's cholesterol. A lot of work in that space. And also having a role in lowering accumulation of central body adiposity. So hepatic is liver, visceral is around the center part of our body, so around the organ. So lowering that accumulation of fat. So that's some of the data that's out there relative to effects of fermented soy products. Anybody know what these are? There's all kinds of fun names. So thinking about dairy beverages, so these are called Kefir Grains. And so just like in kombucha, we talk about that SCOBY or the mushroom, right? These are Kefir Grains, and they are a collection of various bacteria and yeast that come together in these tiny little grains. And this is what is used or added to, it could be a sugar water or to a milk product. So these are the things that we utilize when we were making kefir. Okay, so generating bioactive compounds. Now this is an area that's pretty near and dear to my heart. But it depends a lot on what cultivars, and cultivars in this way, I'm talking about plant variety, what strains of microbes are used, different methodologies, so time, temperature, substrates. And then of course, if we're looking at bioactivity, we want to verify the activity. And so just as a kind of a researchy type of example, because we're all here to learn in Alumni University, if we had the same microbe, 3 rice bran varieties, we'd get a different metabolite profile. Okay, and so this is basically, what are those metabolites that are formed after the bran varieties have been exposed to the same microbe. So what is the role of the rice bran variety? And if I can get my computer to do its thing, I will show you. In this particular example, this was a study looking at cancer lymphoma cells in an in vitro study, and they had completely different effects. And so one of the things that you can do in this sort of setup with a really well laid out plan and a screening assay is to determine which microbes, which bran varieties, what are the conditions, and what metabolites produce the bioactivity to suppress lymphoma cell initiation, perhaps differentiation, etc. This is another one, just an example, red ginseng root, where there's a number of saponins that are in the root itself, but under various conditions. This is just a picture of the chemistry there. You can generate 50 plus different ones, depending on how you ferment the red ginseng root. And through screening assays, this one's actually looking at glucose metabolism, you can find a very specific combination. So the microbe that liberates a metabolite, so a bioactive compound that then can have an effect. And this was an animal study looking at reduction in blood pressure. Glucose. Red cabbage. This is an example of when we ferment it, maybe we would decrease content of bioactives. So we work a lot with anthocyanins. These are pigment compounds. These are the red, blue, and purple compounds that are found in berries, but they're also found in red cabbage, and structure is shown there on the left. And here, when you ferment it, one of the things that happens is that the anthocyanins that show up in people's plasma decreases. So the bioavailability actually decreases. So is this a bad thing? Or maybe there's an increase in other compounds. And so in this particular study, this is not our study, but this is a study that we could do. We could look for other metabolites that increase due to the fermentation process. So while some bioactives may decrease, there may be this entire new population of metabolites that increase, that have biological activity. So this is just a little bit on phenolics. We do a lot of work on that. An area that we're looking to get in a little more closely is looking at Exo Polysaccharides. This will be my last example, and then we'll go into questions. But these are fun components. They're microbial polysaccharides. They're relatively high molecular weight carbohydrate polymers, so carbohydrates that are linked together. There's three main types. There's this lipopolysaccharide type, the capsular polysaccharide type, and then these exopolysaccharide types. And these are the ones that I'm interested in. We do have interest in LPSs as well, but the EPSs are interesting in that they have a lot of diverse functions. They provide protection against adverse environmental conditions. They help adhesion, cell-to-cell interactions, which is important, particularly when we kind of think about physiology, cells talking to one another, biofilm formation, and also serve as carbon and energy reserves. EPSs are produced by a wide range of organisms, plants, algae, but bacteria. Bacteria in specific types, so the lactic acid bacteria, in fact, many of the lactic acid bacteria produce this dextran and xanthan gum. And you probably are thinking, okay, well, what's the big deal about that? But these are functional ingredients that are used a lot in the food industry. You can look at some of your food labels and see xanthan gum for sure, but they give various textural properties to foods. So like yogurt, salad dressings, syrups. I'm showing you a picture of ice cream. Certainly that mouthfeel comes from dextran. It also reduces crystallization. So here, where we can use bacteria for not only what might be good in a specific product that's made, but now to develop an ingredient that has functional capacity. The other thing that's fun about these is that these lactic acid bacteria, which are the main, bacteria used in fermentation, is that when these EPSs are generated, they act as a prebiotic. So if we think about using lactic acid bacteria or certain types of lactic acid bacteria in yogurt, it gives it its characteristic texture and flavor. So it adds that viscosity, okay, the textural properties that we know and love, right? So improves rheological properties because it also keeps things from this kind of weeping. But then on top of it, these EPSs can feed those probiotics or those bacteria once they're in the gut. So in this particular case, we have functional ingredients ×2 with these Exo Polysaccharides. So choosing the bacteria that has these multiple functions, including making these EPSs, can set up this symbiotic type relationship for foods that would then have beneficial effects. So I think it's an exciting area to look more into. Hasn't gotten a lot of attention, but I certainly think that it's an area that deserves much more attention. And just to give you an example, this comes from a paper, actually a review from not too long ago, looking at bacterial EPSs and their health benefits. The center of this particular table shows you some of the effects that have been documented, but not too many documented in humans, unfortunately. So wide open area for us to look further. So my final slide, we all ferment. We have to ferment for life. We generate ATP when we have lots of oxygen, but if we don't have oxygen, we have a pathway in which we will make lactic acid, and that's kind of shown down here, when we're stressing those muscles and we've run out of enough oxygen, and so we have this other pathway, it's the lactic acid. But, we look at fermentation in so many other parts of what we do, where we live. On the left-hand side here, you can think about composting. Those of you that love wine and beer, certainly it's getting to be about that time. If you have a cocktail hour, we use bacteria fermenting all kinds of really beautiful beverages, both ethanol containing as well as not ethanol containing. And of course, we have the various processes that I'm showing here relative to some other food products, dairy just being one of them. So with that, I will end and take some questions. I hope that gave you everything you wanted and more about fermentation. I think you can appreciate that while it's an old process, it's really coming into a new age where we understand it and utilize it differently. And we'll get a lot of culinary pleasure out of it, but also hopefully some long life out of it as well. So thank you very much.
Thank you so much, Britt. We do have some questions. I will turn it over to Meg to ask the first one. And as a reminder, if you have questions for Britt, please feel free to type them in the chat box at the bottom of your screen, and Meg and I will make sure that they are answered.
I also have my email there in case someone comes up with a burning question after and don't have the chat box to put it in. I'm happy to take questions.
Excellent. Thank you so much, Britt. As Kristen mentioned, this was so fun and educational, and I never realized how many things I actually consume that are fermented. So I'm excited I got to learn. One of the questions from the chat was, how does gut microbiome develop in babies, and at what age does this happen?
Oh gosh, what a great question. Okay, so in babies, they are colonized. Their colonization begins at birth. And so cesarean delivered babies compared to vaginal delivered babies will have a different start, if you will, to what microbes will be there first. And then mother's milk. So breastfed babies, there are some phenomenal oligosaccharides that are found in breast milk that help to form that the baby's gut microbiota. Infant formula will also feed the gut microbiota, but it will have a different composition. There are distinct differences between babies that are born vaginally and by cesarean, and there are distinct differences between babies that are formula fed versus babies that are breastfed. By the time they start getting on to solid food is when they start developing a more diverse microbiota. Their microbiome starts to look more mature because of the different foods that they're consuming. I'll add that an infant formula fed baby tends to have a more diverse microbiota than a breastfed baby. And that's one of the differential features of the two. And so by the time they're 3, 4, I mean, they've been on solid food for a while. They're looking, they're not adult-like, but they're certainly looking more adult-like than they do when they're infants. Does that answer the question?
I think so. Thank you.
Mm-hmm.
I know, Susan,
Oh.
you had a question.
Yes. So our next question was about how could fermented foods be used to help areas with food insecurity or in food deserts?
Yeah, great question. In fact, one of the things that fermented foods is that it helps increase shelf life. We can utilize raw materials that are not as expensive and create wonderful products. There is some, how would you say, not everybody likes the flavor of a fermented product, right? And so there is some acclimation to that flavor. And so, a lot of the foods that we consume that are fermented usually come through, experience, and that usually stems back to what they grew up with. People that are exploratory in what they try and taste certainly will develop a more diverse palate. But indeed, it could definitely be utilized to help increase availability of a wider selection of foods. I do worry a little bit about just the acceptance part of it, depending on the flavor profile, but it definitely could be utilized.
Thank you so much, Britt. I think our next question was from Jason. Are there any food products that are fermented in both preparation and in the end phase? And if so, what benefits could double fermentation have?
Trying to think about in the beginning and in the end. So like you would have it done in the beginning, you'd have more something going on, and then kind of to finalize it. So I can't really think of anything right off the top of my head, but I can tell you there is this movement of, and kombucha is one of those areas where they will do the fermentation, they'll pasteurize, and then they'll add back live microbials, very specific live microbials. There's a lot of research that's being looked at in that way to see if you can get all the effects of a more comprehensive fermentation process. So more microbials, more microbials in the early phase, and the components, the bioactives that might be generated, stop that, and then add back the microbes of interest, maybe more as a kind of the probiotic effect, so that you have those very specific microbials that would, then, be important for gut health. So that's the one example I can think of. I'm sure there's going to be a lot more. There's interest in, shelf life for sure, in, how long or how much, from a, number standpoint, some of these stay alive, and is there a way to enhance that? But again, that's really more in research and development right now.
Thank you, Britt. Another question came in regarding kombucha. And the attendee has asked, how do you choose a kombucha? I guess they have read that some can cause some type of skin allergy or rash. Can you maybe talk a little bit further about that?
Yeah, well, I can't talk a lot more about it because it's a bit of a trial and error right now. This is an area that definitely needs some regulation, needs some standardization. There have been improvements, as I said. You know, it's the industry has come a long way, but it's not entirely clear why some might give somebody a reaction, whereas another type may not. And that's where some of the standardization and expectation of products. So I really don't have a good answer for you right now. But for some people, they have a hard time tolerating it. And it may be something that is not worth going towards at this point, and waiting a little bit. Nowadays, commercial kombuchas don't seem to be quite as risky, if you will. Homemade kombuchas could be a little more risky in that regard.
Thank you, Britt. So we have a question here. Given the difficulties of conducting human studies, what are the best animals to use in preliminary research? Can plants be used for these studies in fermentation?
So turns out we don't actually think human studies are hard to perform. This is what we do every single day. Animal studies, your model would be chosen based on the health benefit outcome that you're looking for. So for example, if you are interested in a cardiovascular disease risk factor, okay, or risk factors, you might choose a certain mouse model or a certain rat model that is designed to test some of the risk factors for cardiovascular disease. Likewise with diabetes, okay? It is hard to think about, just because of the differences in plant metabolism, how we would be able to utilize plants to study biochemical, physiological reactions that would then depict what would go on in human physiology. So I don't really think that we could probably use plants. There are some methods that are being developed relative to taking different cell types and putting them together to simulate what might go on physiologically. So it's called like gut on a chip or organs on a chip. And so you might have on a chip cells from the GI tract, gastrointestinal tract. You would have cells from the liver, cells from the adipose, cells from the immune system, so that you can then study some level of interaction that would give you a sense of how certain maybe fractions of a fermented product might impact these cells and how the cells interact in response to, you know, that fraction. So that would be more of a screening assay for biological activity and maybe studying mechanism of action. But that's probably kind of the latest and greatest as it relates to not using animal models, not using human models, and having something that, you know, we could, you know, really take back and say, all right, this has some connection to human physiology. We have some really good information from it, and now we can move forward.
Thank you, Britt. And we have time for just one more question. Someone has messaged in, do the bacterial/yeast species on Kefirs form a community that has a consistent composition over time, or do they change?
So I missed so a bacteria or yeast on what?
On Kefirs. I guess the
Kefir grains?
The Kefir grains. That's what I'm assuming they mean.
Oh, okay. Do they change over time? So certainly depending on how they're formulated and how they're fed, I would assume so. I don't know how much they change, but yeah, it's a living system, right? So it would make sense that they would change. And that would be more compositionally and based on the substrates that keep them alive.
Well, thank you so much, Britt. We are right at 6:30. So if we have any further questions, Britt has included her email on the screen, and we will also have this presentation available at iit.edu/alumni university in the next, hopefully week or so. So thank you again, Britt. This has been absolutely wonderful. Just a reminder to everyone that our next Alumni University presentation will be on February 16th from 5:30 to 6:30 p.m. And we will have Sarah Hansen and Matt Callione, who are landscape architects, discussing their work in landscape architecture and urban environments. So we hope that you all can join us for that one as well. So have a great night and thank you again.
Right. Thank you so much, everybody, for joining. And thank you, Kristen and Megan, for putting this on for us. It was really great.